Yep, and if I see them, I'll try and answer them there and then. If not, I'll try and get back to them after the talk. So my title of my talk is, firstly, thank you, Hamish and Kevin, for having me here. It's wonderful to be here, to be able to present locally as well to my fellow Blue Mountains people, and for all those who are tuning in away from the Blue Mountains, hello as well. So I'm going to talk about cell membranes, which is something I got into when I started work at the Victor Chan Cardiac Research Institute when we moved to Sydney in 2010. And it's taken me all the way to UTS, and now I'm part of a lab that's specialised in cell membrane research. So if I can just get my slide to transfer, there we go. So cell membranes, as hopefully most of you will know, that around every cell, all species, have a cell membrane. And it's a very, very thin membrane. It's only about 4 nanometers thick. It's very, very thin. It's impossible to see under normal microscopes without some sort of contrast agent there to highlight it, because the thickness is smaller than the diffraction limit of most optics. And so some species, like plant species, will have a cell wall as well as a cell membrane, and bacteria also have things like a lipopolysaccharide layer and may also have their own walls as well. And some bacterial species actually have 2 membranes, a plasma membrane plus an outer membrane as well. But for mammals, we have just one membrane on our cells. Oh, viruses also can have these little envelopes, which is like a membrane. Some viruses have them, including the coronavirus. So the good thing about cell membranes is that they keep things in one space, located in a very, very small space. So that stops chemicals and reagents from floating away, and it keeps them concentrated. So various reagents and chemicals can find each other when they need to react with each other. So they're very important in that regards. Another thing that's important is that the cells are kept kind of small. And so there's a theory that says that cells can't get too large because the surface area to volume ratio of that cell changes such that the volume increases at a greater capacity than the surface area of the cell. And so you can't get enough stuff coming in to feed the cell enough, as it were. So that's why your cells are small. And that's why we need an awful lot of them to create something as big as an elephant or me or whatever. So that's a brief introduction. Let's go back in time. And people didn't realize there were cells until they started mucking about with optics and created the first microscopes. And the guy who termed the coin cell was this guy, Robert Hooke, who came up with the word cell. And he was looking at cork, a plant species, and he could see these various little windows, if you like, or what he called them, cells. And he could also see that they had a cell wall, because it was a plant. You could very clearly see they had a cell wall, but he couldn't see there was a membrane there. But then if we look at mammalian cells, because we can't see there's a membrane, we can distinctly see down a light microscope that there's individual cells there. But what's holding all the stuff in? What is it that's holding all that stuff in one place? And that was tricky, and that wasn't actually solved until the 20th century. And part of the discovery had to do with anesthetics. So Hans Meyer and Ernest Overton separately noticed that the potency of anesthetics increased by the more they like separating from water into oil. So the more they like anesthetics like going into oil, the more potent they are. Now they both figured this out separately. In Hans Meyer's case, 2 years before Charles Overton, but he published his in German, so probably they didn't get translated in time. And then Charles Ernest Overton came along, more commonly known as Ernest. And what Ernest also thought was that because that's the case of anesthetics, maybe cell membranes are made of oily stuff. that anesthetics can dissolve into, e.g. lipids. And that became known as the Overton biomembrane model. And just as an aside fact, we still don't know how anesthetics work. We've got ideas. Is it something to do with the membrane? Is it something to do with proteins in the membrane, proteins associated with the membrane? But different anesthetics work differently is now the current thinking. And is it membrane, how important are membranes in that? We still don't know. So we still put people to sleep without actually knowing what's going on when we put them to sleep. Anyway, that's an aside. Oh, I need to make a diversion because right now I need to talk about water. And you'll see why I need to talk about water in a sec. Because water, as we know, is H2O. We have 2 hydrogen atoms here in the white and this red guy here, which is an oxygen atom. Now, oxygen, if you remember your chemistry, has a 2 negative charge typically, and hydrogen has a 1 positive. So you have 2 positives and overall 2 negative here. And so overall, water is a neutral molecule. It's well balanced. It's got 2 pluses plus the oxygen, which is a 2 neg, come together, they cancel each other out. However, because of the arrangement of the electrons in their shells, the hydrogen atoms primarily like to reside in this kind of formation with respect to the oxygen atom. And this means that there's no symmetry here. And so one side is where the electrons tend to hang out more than the other side overall. I mean, they're wiggling and jiggling about, all the electrons are buzzing about madly. But overall, the probability is that the electrons will hang out slightly more, spend more time on this side of the molecule than this side of the molecule. And this makes water slightly more negative on one side and slightly more positive on the other. Even though overall it's neutral, the molecule's neutral overall, it does have this polarity. So water is what we call a polar molecule. Okay, what's this got to do with cell membranes? I'm getting there. As we know, opposites attract. And so when water, in our glass of water here, water is jiggling away, jiggling, 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 jiggling. And the opposites, the negatives are being attracted to the positives and the positives are being attracted to the negatives, and they're all moving about each other all over the place. And so they'll congregate together. They like to be together because they've got opposite charges in which to attract that are attracted to each other. But there's other molecules that are non-polar. In these molecules, typically with carbons in the middle and hydrogens on the outside, the electrons are shared more evenly in these molecules. And they're of a symmetrical shape as well. And so these molecules tend to be non-polar. They don't have this electronegativity associated with one side of the molecule compared to the other. And that makes these particular molecules quite hydrophobic. They're scared of water, which is literally what hydrophobic means. And so oil and water don't mix. If you put them together, if we put our water molecules here together with our oil molecules, all the water molecules will like to congregate together and they will squeeze out all the oil. And so if you put oil in water, that's exactly what happens. The waters all want to congregate together because the opposites attract, but there's no opposites attracting in the non-polar molecules, and so they get separated out, squeezed out by the water molecules. Okay, I'm coming back to lipid membranes, the cell membranes. There's another class of molecules which are called surfactants, and they have polar and non-polar moieties attached to them. I shouldn't have used the word molecules there. I should have used the word moieties, excuse me. Anyway, so here is what a classic surfactant might look like. Up the top here in the color, in the red and the white, is the polar part. And the water molecules here are attracted to that. But down here is the non-polar part. And the water molecules don't want nothing to do with it. And so these sort of molecules are also called amphiphiles. They're amphipathic. And so these are the surfactants. And so they have a hydrophilic top portion, or one portion of their molecule is hydrophilic and the other portion is hydrophobic. Now we can draw this, well, what we like to do is draw it in a simpler form. We'll just put a big circle here around what we call the head group, the hydrophilic bit that likes the water. And we need to include the waters here because that expands the size of it a little bit. And we'll just make this stick here representative of the hydrophobic parts. And so we'll call that our surfactant, our amphiphile. Okay, so we'll come back to cell membranes in a sec. This guy on the right here, Irving Langmuir, he thought, these surfactant-like molecules, if I put them in, if I added them to the surface of a bath of water, then their hydrophilic parts, the parts that like water, will stick into the water, but the hydrophobic parts, the parts that don't like water, will have their tails sticking up. And he and Catherine Blodgett used this device called the Langmuir-Blodgett trough. Incidentally, Catherine Blodgett was the first woman to get a PhD in physics from Cambridge University, if you didn't know that already. But anyway, they came up with this thing here called the Langmuir-Blodgett trough. And what it does is it puts these surfactant molecules onto the surface of water, and the head groups, which are water-loving, will sit in the water just at the interface, and their tails, these hydrophobic portions, will stick up out of the water. And this happens. This happens in the ocean. Right now, on top of the ocean, there are little surfactant molecules sticking up with their tails sticking up outside of the water. And there are certain classes of microorganisms that live very close to the surface of the ocean that can feed off these molecules. Anyway, that's a different story. Come back to the Langmuir-Blodgett trough. So they put their lipid molecules, their surfactant molecules, onto the surface of the water, this bath, and then they got this barrier here on this side, and they pushed it in, and then they measured the surface pressure. And so they squashed all these lipid molecules, these surfactant molecules together, and they got a lovely shape, a plot going up and up and up, but then it kind of changed shape. And where it changed shape is sort of where they got compacted enough so they won't start overlapping and piling in on each other. And about that point there, they can measure the surface area, and from that you can get an estimate of how many lipid molecules you have. Okay, well that's a good technique, this Langmuir-Blodgett trough. So these two Dutch scientists then came along in 1924, and they got all the lipoids, all the surfactant-like molecules. They isolated them from red blood cells from different species, and they knew exactly how much they had, and they put it onto a Langmuir-Blodgett type trough on top of the little water, and they compressed it, and they measured the surface area of their compressed little lipid layer, and they figured, oh, there are twice as many lipids as would be required just to make a monolayer. And so they went back and thought, okay, we've got the cell membrane components from a red blood cell. We've separated them out. We've put them on top of our layer. We've squashed them. Now, if it was just one lipid molecule, then we should have this many lipids in this surface area. But there were twice as many. And so that made them think, there must be a bilayer being formed. So there must be enough lipids here and enough lipids here to form a bilayer. Now, their experiments weren't very good and they got a few things wrong, but they just happened to be right. When it came to cell membranes, it is a bilayer. There are two layers going on. And electron microscopy finally confirmed this sort of stuff. There is a membrane around cells that's keeping stuff in. But electron microscopy, though, still can't tell you if it's a bilayer or not. That could just be one layer, for instance. It could be three. But the early experiments seem to suggest it was a bilayer. But what are these oily lipid molecules that make up our membranes, and what specific properties do they have. Okay, so we go back to our surfactant. And it's got a hydrophilic side and a hydrophobic side. We've created that shape to describe it. Okay, that's a nice shape. Let's take that shape, and we'll put it in the middle of our screen so we can see it better. And if we look at it in terms of volume as well, we could probably create a sort of a cone shape around it. That's its overall shape in 3D. Now, if we were to stack these together, this particular surfactant, this particular detergent, if we were to stack it together, it would form that shape. So this particular surfactant here with its one tail, its head group, has a kind of a cone shape. And if we stack them together, it would look like that. And that's what happens, and that's what we call a micelle. So in a micelle, all the surfactant molecules have a hydrophilic outside and a hydrophobic inside, and they form this micelle. And where you get them in particular is in your dishwashing detergent. So we make use of the micelle, we put it in our detergent, we mix up our water, and we get all these little micelles created. And those micelles can then become home for the fats and things on our dishes. So the fats can have a place to go to in the water, inside the micelle, where they're protected from the water, while the water is protected from it, because the water is scared of fats. Fats and water don't mix, right? So the fats need a place to go. And so that is what we use in our dishwashing liquid. We create these micelles. And it also really eases the surface tension on the water as well. But that's a different story. I'm not here to discuss washing the dishes. Okay, so that's a sort of surfactant that has a kind of a one-tailed lipid. So we call it a one-tailed lipid. What if we had two tails on our lipid instead? Two hydrophobic portions of our lipid. Well, in that case, it would have a cone shape. Cone shape. And so if we have a cone shape, what forms instead is not a micelle, but that bilayer I was talking about. We'll have a hydrophilic outside, hydrophobic inside, where the chains are, these chains here, we call them. And then the head groups again with a hydrophilic outside. And so what we have created is a bilayer, a lipid bilayer. And that's what cell membranes are made of typically. And so with a lipid micelle is what we discussed. If you've only got one tail and it kind of forms a cone shape. If it's got two tails, we can form a lipid bilayer. And those lipid bilayers themselves can join up and form their own sphere. And we call that sphere a liposome. A liposome. If we're using it in the lab, we can make liposomes in the lab by using certain types of lipid, putting them in water, putting them through a filter, and we get liposomes formed. And liposomes are very cell-like structures. And it's more energetically favorable for lipid bilayers. So up the top here, we have what's called a planar lipid bilayer, a kind of flat lipid bilayer. But if you put it in water, this flat lipid bilayer, it's more energetically favorable for it to form into a liposome, into one complete cellular liposome. And often they can form into multilamellar liposomes, where you have lipid bilayers around lipid bilayers around lipid bilayers around lipid bilayers around lipid bilayers, and they can be multilamellar. What we like to work with in the lab in particular is unilamellar, meaning just one bilayer forms this wonderful sphere, because they're good to experiment with, because they look and act a bit like cell membranes would, a bit like cells would. What, however, if we have this detergent here, with the part that likes water quite small compared to the part that dislikes water? In that case, we get this kind of other shape, and if you pack them together, we can get something called an inverse micelle, where the hydrophilic portions, the parts that like water, are in the middle, and the hydrophobic portions are all on the outside. That's called an inverse micelle. And so it depends on the shape of your detergent molecule, how things form. And for cell membranes, it needs to be more of a, overall, it needs to be more of a cylindrical shape. You can have these other shapes in there. You can have them squeezed in there, but overall, it needs to be, the majority of lipids need to have a more cylindrical shape in order to make that kind of cell membrane. And so a typical cell then will have all these lipids, most of them two-tailed lipids, and they're all jiggling about. It's quite fluid. Things are moving about. All these lipids are moving about around each other in their own little bilayer, in their own leaflet of the bilayer. They're moving about each other. And always the head groups, the hydrophilic head groups on the outside and the hydrophobic portions on the inside. And in 1972, the model of a cell membrane, which is still used today, which is still the most common model and most widely believed still, is what we call the fluid mosaic model of cell membranes. And in this fluid mosaic model, the membrane is quite fluid, the lipids are quite fluid and moving about around each other, going backwards and forwards in their individual two leaflets. And we get proteins and other large things sticking through the membrane. And sometimes these proteins are attached to anchor molecules within the cell, such as cytoskeletal filaments. Sometimes they have bits sticking out of the membrane, such as glycolipids, which have sugar molecules sticking out of the membrane. Sometimes they form ion channels, which form pores in the membrane, so ions can travel in and out of the membrane. So this, a good example of a protein channel. Where's my zoom gone? Here it is. This guy here. So that's what we call a protein channel, an ion channel, and that lets certain ions in and out of the cell. And other proteins are there to traffic other things. So they can grab a molecule and help transport it through the hydrophobic parts of the membrane and into the cell. So there's lots and lots of proteins that can be associated with our cell membranes. Okay. So what about the types of lipids in cell membranes? There's lots of them, and here's just a kind of a pictorial overview, if you like, of some of the lipids. So if they've got straightish tails, hydrophobic portions are kind of a straightish tail, they're called saturated because they contain no double bonds. If the molecule had a double bond in it, then it would be unsaturated. So if you do put a double bond in, like this one, then often you get a little kink in your tail. And that has properties, that changes the membrane properties. If you have a lot of lipids with these kinks in their tail, then the membrane becomes more fluid, whereas these guys are more rigid. You can have asymmetric tails, very common, where one tail is longer or shorter than the other. And you can also have lipids with different head groups. So the hydrophilic portion at the top that associates with water can have different head groups, such as a choline head group or a serine or a glycerol head group. And some of these can be positively or negatively charged. So in nature, you get negatively charged lipids, such as phosphatidylglycerol, and they're typically in bacterial species. Positively charged head group lipids, well, they're only made in the lab. You don't find them too much in nature. Most mammalian cells have a kind of a neutral charge, though. So they're called zwitterionic. But bacterial membranes in particular have negatively charged lipids. And we'll come back to that in a sec because that could make them a potential target for certain drugs. And then there is sterols. So they're not phospholipids. The other two-tailed ones I was talking about are called phospholipids. These guys are called sterols, and they've only got a tiny, tiny, tiny little hydrophilic part, and then a much larger hydrophobic part. And so cholesterol here is your classic example of a sterol amphiphile, but it's not a phospholipid like the other molecules I was talking about. And so their shape would be tiny little hydrophilic portion and a much larger hydrophobic portion, but it's not a very big molecule compared to some phospholipids, and so it would sit in the membrane next to them, but only going so a little bit, not so deep. And they reckon cholesterol is there to make the membranes more rigid. So if you start, if these lipids here, excuse me, are too freely floating around, cholesterol in there is to stop them moving about and make them a bit more rigid. But cholesterol can also hang about with itself, as can other lipids in the membrane. They don't necessarily stand next to each other. And you can get lipid domains occurring. And here's an example of some very smart scientists who managed to color certain lipids. And if it's just one sort of lipid, it's all very homogeneous. But when they put in two different types of lipids and color one, you can see they've formed all these wonderful circular domains in their liposomes that they've created. And that's the lipids, what we say, phase separating out into their own little domains. They like to hang together with each other, but they don't like to hang out with the other lipids in the membrane for whatever reason. Okay, excuse me for a sec while I take a drink. And so also, lipid packing is very important as well. And lipid packing can determine what the membrane is like and how it functions. So often at lower temperatures, lipids become more compact together. There's not a lot of energy left, and they don't like wiggly jiggling around anymore. They want to sit still and be in one spot. And often lipids without double bonds in them, so very straight tails, will be able to pack together closer. And so they become rigid and tightly packed. And we call this the ordered or solid phase of lipid membranes. Alternatively, if we have lipid tails that are less ordered, they're more fluid and able to wiggle about and get in between each other and get around the place. And increasing the temperature of your membrane can also cause that. And that's exactly what happens with butter, for instance. Butter is in the solid phase at low temperatures, but as we all know, when we increase the temperature, butter starts to melt and goes into the fluid phase. Another good example might be candle wax, where solid phase, sticking together, heading into the liquid phase, happy to move about. And cell membranes are a bit like that as well. You can have regions in the cell membrane that are quite packed, solid, and you can have other regions which are quite fluid, moving about. Okay, so that's a bit about the background science of cell membranes. Let's talk about some of the sort of research that we do in our lab, in the membrane biophysics lab. And I'd like to introduce you and say hello to Alvaro here and Evelyn, who are also leaders in our lab. And we work together on a whole lot of projects, often to do with the creation of membrane pores, in membranes, and various things that can help create those pores, as well as doing some various pharmacological research as well. Evelyn in particular, she is the master of molecular dynamic simulations. So what she can do is she can model in a computer a couple of hundred lipids and create a lipid membrane in a computer in silico. And then she could add various proteins and see where they fit and how they insert into membranes in silico. She can do dynamics where you add something and then you wait and see what happens and where it goes into the membrane. And this is all to do with supercomputing. So this needs supercomputers down in Canberra somewhere, which she logs into from her laptop and then sets that up, runs the experiment for six weeks. And it's an all atom experiment. And so it takes a whole lot of number crunching to happen. And then maybe, if we're lucky, we'll get about 1 microsecond's worth of data, of 1 microsecond's worth of time data of what's happening in that membrane. These all atom experiments take a huge amount of grunt, and that's what Evelyn specializes in. So that's one sort of experiment that we do in our lab. Another important experiment that we do in our lab is called a tethered bilayer lipid membrane, or a TBLM. Now for this, I'm going to just duck out and go to this presentation here. Hamish or Kevin, you can see it?
We can see it. Looks fantastic.
Thank you, mate. Thank you. Okay, so here I've got 2 gold electrodes. The top one, the big one, that's just our return electrode. And for the rest of this talk, I'm going to ignore it because it's boring. But down at this bottom electrode, I'm going to add some chemicals. I'm going to add 2 sorts of chemicals. I'm going to add this little guy here, which we call a spacer molecule. And then there's this bigger guy here, this bigger chemical, which we call a tethering molecule. And this brown part here, it's got chemistries that are hydrophobic. So this part here is hydrophobic. All these other parts here are hydrophilic. Down here in the yellow, these are sulfurs, and sulfur likes to coordinate with gold. So it's just attaching to gold. It's going to stick on the gold, and we can't get it off once we put it on. And so I'm going to cover my gold electrode with all these chemicals, these tether molecules and these spacer molecules. And those spacer molecules there, they're there to hold the tether molecules up so they don't all fall over. And then what I'm going to do is I'm going to bring in some lipid molecules. And when I bring in lipid molecules, phospholipid molecules, I can create a membrane anchored onto my gold electrode. And it's anchored slightly off this gold electrode so that if I add something to this membrane, there's a bit of space just between the membrane and the gold electrode for things to sit in, for ions to sit in, or for little bits of protein or whatever. And so these hydrophobic portions here are associating with the hydrophobic portions of the lipids in order to help create this lipid bilayer. So it's a model cell membrane we've created on a gold electrode. And we'll cover our electrode with our membrane. And if I wanted to look under it on a microscope, this is what it would look like. And so we've added a fluorescent dye into our membrane here so we can see it. And we've scratched away some of the membrane, and it was hard to scratch away. So really, we've really well coated this electrode with a lipid bilayer, and that's very useful for experiments. So what we then do is we create a wash path, a path through which we can add chemicals, and then because we can add a circuit to it, we can use something called electrical impedance spectroscopy to measure how things are interacting with our membranes. And we'll wash through chemicals here and measure what's happening to our membranes when we wash through those chemicals. So that little wash path there just looks like this. And so here's a slide. And each one of these electrodes here is where we've formed a tethered bilayer. We've tethered a bilayer onto our electrode. And this little cartridge here, which we get from a supplier, an Australian supplier who we work very closely with, plugs into this little box here, which is called an electrical impedance spectrometer, and that just feeds into our computer. And then we can add things to these little holes and wash them across our membrane and see what happens. It's a very, very cute technology. So that's tethered bilayers. Now, Hamish, can you see back to the screen now? Back to what?
All good.
All good? Cool. So we create these tethered bilayers on our electrodes, which fit on a cartridge, which plugs into this little box. And that little box is very portable. We can take it anywhere we want to take our laptop pretty much, because it just plugs in with a USB port, and we can measure how things interact with membranes. So what are the sorts of things we're measuring? Well, this particular frog here, called the South African clawed frog, it swims in dirty, filthy water, and in its skin, it produces peptides that are antimicrobial. And it's producing those peptides to defend itself. Now, these antimicrobial peptides are positively charged molecules. Now, if you remember back to what I was saying before, bacterial membranes have negatively charged lipids in them. That could be interesting. So, with some collaborators from Germany, I got some of these peptides and I wanted to see if these peptides created ion channels in my, if they created pores in my membrane. And if they created pores in my membrane, I should be able to measure that by a change in membrane conductance that I can measure with my electrical impedance spectrometer. So I got some of this chemical and I added it to my membrane. And sure enough, when I increased the concentration of this peptide on my membranes, on different membranes, I got an increased response, an increased conductance response, which means that it was punching holes in my membrane. Cool. Okay. But what about this membrane here that I was using? That was a neutral membrane, a zwitterionic membrane. So it didn't have any charge. What about if I made a membrane instead that had negatively charged lipids in it? And so if I add a proportion of lipids, that these white ones here that are negatively charged, and I add my peptide again at a fixed concentration, then the more negatively charged lipids I have in my membrane, the bigger the response of this antimicrobial peptide. And so that's just one of the things we can measure. We can look at all sorts of antimicrobial peptides. We can use different lipids, and we can use different lipids to model bacterial membranes, and we can see if these peptides, these antimicrobial peptides, will kill bacteria, or we can figure out how they're killing bacteria, because you could also do experiments on bacteria to see if they do kill the bacteria. And we can see if their potency is related to their membrane effects. And with bacterial resistance now being a problem, the idea is for these antimicrobial peptides to be more impervious to bacterial resistance. Okay, another thing we're doing is we're looking at something called a phospholipase, or phospholipases, I should say. Phospholipases are enzymes that attack our lipid molecules that make up our lipid bilayer. And so they're very important. Your body makes phospholipases. It's important for cell function, for your cell membrane function. And they're responsible for degrading lipids in the membrane, for instance. And there's various types of phospholipases. There's phospholipase A, phospholipase B, phospholipase C, phospholipase D, and they all have slightly different actions. And what they do is they catalyze the cleavage of lipids. So phospholipase A2 here, for example, catalyzes the cleavage of this lipid at this point here where this ester oxygen, this double bond oxygen is. Phospholipase A1, a close cousin to phospholipase A2, attacks the lipid on this particular chain, which is just slightly offset. Phospholipase C will cleave just below this phosphate group in this phospholipid. And phospholipase D will cleave off this choline molecule at the top of this phospholipase, or this phospholipid, for example. So they're phospholipases. So in summary, if I was looking at phospholipase A, we have a lipid, and then what it'll do essentially is just cut this tail off. And so we've got this tail, we'll cut it off, and we'll cut it off where the double bond oxygen is. That's what phospholipase A does. Now, many species, many species, oh, sorry, I'll come back to that in a sec. So we can use tethered bilayers then to measure the actions of phospholipase A. And so we've got these ester oxygens here. It's that's where the phospholipases are going to work, and they'll cleave them. And then we'll have what we call an oleic acid, which is just this one product, and this other product here, which is called LPC. And when you get too much of this sitting in your membrane's going to fall apart. And we can measure that by a change in conductance. We can also create membranes that have ether oxygens, no double bond oxygens, just these guys here. And phospholipase A's then shouldn't work on these membranes if we have these lipids. So we've had these ether lipids instead of ester lipids. Phospholipase A shouldn't work. And that's exactly what we see. So when we have ester lipids, ester phospholipids with those double bond oxygens, phospholipase A is happy to upset the membrane. And you can see that here. When we add phospholipase A to these ester lipids, this DOPC ones, we get a huge response in membrane conduction as our membrane starts to fall apart. But on these tethered bilayers, nothing happens because there's no ester oxygens on which the phospholipase A can work. And so we've created a phospholipase A sensor, which is very handy. Because phospholipases, such as phospholipase A's, are found in venoms of many creatures. So, particularly in snakes, particularly in vipers, you'll get a lot of phospholipase A2. In many ants, in their stings, you get a lot of phospholipase A2. In certain spiders, you get a phospholipase D variant as well. And so we've got a tool then now to look at how the actions of, some of the actions of venoms might work. Now, in most venoms, there's a whole heap of toxins. It's not just phospholipases, okay? But if we want to know whether there are phospholipases in there, or if we want to try and get a fingerprint of these venoms by the amount of phospholipases in there, then perhaps we might have a tool to do that. So this particular very intelligent scientist named Dr. Samira Aili, who's now going on to do medicine, she once did a proteomic analysis of bull ant venom. So this is the South American bullet ant. And she thought, by looking at the proteins in it, oh, this protein looks like it might be a phospholipase, but we won't know until we test it. So she did a proteomic analysis. She looked at all the proteins and discovered that, oh, it's probably phospholipases in there. Now, this particular bullet ant, this South American bullet ant, gives the most potent sting you can imagine. You've probably heard of it. So Hamish, Hamish Blake, the very famous Hamish and Andy, went to South America. Went to South America and let himself famously get bitten by this bullet ant. And he was shrieking in pain for ages. And if you go to YouTube, type in Hamish bullet ant, there's a video of it from their TV program, their Hamish and Andy TV program. And you can see what sort of pain Hamish was in when he let these ants bite him. Don't let these ants bite you. He ended up in hospital, of course. Wasn't very nice. Back to the story. We want to see if there's phospholipase A in there. And so that's what we did. We added our venom from this particular ant that we got with some collaborators. And we saw in membranes with the ester lipids that we've got a huge response when there's calcium in the solution. There's only not much of a response when there's no calcium. And it's well known that many phospholipase A's need calcium in the solution in order to work. Now, unfortunately for us, we've got a lot of calcium flowing through our veins, so it's going to work in us too. But when we use the ether lipids without the esters, it didn't work at all. And so we could show that this particular ant, for instance, had phospholipase A2 in its venom. But there's lots and lots of other things we can use this particular kind of sensor for. So your pancreas, for instance, releases phospholipase A's into the gut when it wants to dissolve, when it wants to break down the fats that you eat. And sometimes it also releases this other stuff, other lipases as well, which aren't phospholipases, but they do break up other fats. And we can create tethered bilayers of those other fats as well. And we can use that as a sensor for lipase activity. And that can become important with diseases such as acute pancreatitis, which is an inflammation of the pancreas. And when that happens, the cells start releasing too much of these enzymes and they get into the bloodstream. And if we take some of your blood of someone suffering from acute pancreatitis, we can say, oh, look, there's a lot of lipase activity. It's acute pancreatitis, quick, get them to hospital. And they do need to go to hospital because it's quite a serious condition. And particularly in remote settings where there's often a high incidence of acute pancreatitis, often because of alcohol, that becomes a very important diagnostic. But PLA2 is also important in other inflammatory diseases, such as ulcerative colitis, which is like an inflammatory bowel disease. And we've got some very exciting data showing from healthy patients and patients with ulcerative colitis. And we looked at their stool samples and we measured their phospholipase activity. And I can't go into any more, but the data is exciting. I'm not allowed to say any more because of, people want to make money reasons. So But that's exciting. That's one of the ways we can use our phospholipase sensor. So what else is our lab doing? Well, we look at spider venom peptides, and we look at their capacity to target cancer cells. So this is something Evelyn's doing. Cancer cells have a lot of a particular lipid called phosphatidylserine in them. And she's looking at peptides that target the phosphatidylserine lipids only, which is quite exciting. And we can use our tethered bilayers, and she can use her molecular dynamic simulations to do analysis of that. Evelyn's also looking at antimicrobial properties of honey, particularly manuka honey, because it's been shown that manuka honey is very bactericidal. And so we're taking apart the components of the honey and seeing which components in that honey are the actual parts that might interact with bacterial membranes. Cancer cells also often live in a more acidic environment. And so we can test peptides that might insert into membranes and punch holes in membranes and kill cancer cells. That will only work in the acidic environment, but not in the normal neutral environment that most of our cells live in. So that's another field of research we're looking into. And Alvaro, our other colleague, he's looking at statins. Now, statins are cholesterol lowering drugs. And so cholesterol, and there's lots of theories about how it might interact with membranes, these statins, and whether their effect is just in the production of cholesterol or if there's another effect to do with membranes as well. So that's another field of study that we're going into. And we're also looking at general anesthetics. We want to solve that problem. Is anesthesia, is the narcosis effect due to what's happening in membranes? Is it due to lipid membranes? And we've got some pretty good data about that as well. But that's a side project for us at the moment. I think I'm coming towards the end of my talk now. Oh, and that being the case, I'd just like to give a shout out to any of my P&C colleagues from Springwood High School. And I'd like to point out too, if you can all go along to Facebook and go to this Too Hot to Learn site and give these kids a like, because these kids there, they do their work at a high school with no air conditioning. Every government office in the state, I'm sure, has air conditioning, but why can't our kids at high schools or our teachers at high schools have air conditioning? And with the climate changing as well, you might have seen these kids on that TV show, Big Weather. Jump on iView and take a look if you haven't. And if you want to, you can support these kids by just going to the Facebook page and giving them a like. I've got to declare that my daughter might be one of these kids, maybe this one here. So please go to Facebook and give them a like. Also, just for more the local people in the Blue Mountains, Operation Bike Shed, where we're converting this container into a bike shed for Springwood High School. We've opened it up now, we've put windows in this container, and we're ready to start painting. Go to our Facebook page and look for announcements, and hopefully, weather dependent, next Sunday, I'll be looking for volunteers to help paint this thing. So if you're free, please come along. I need to thank my fellow researchers, of who there is a long list here. I need to thank my funding bodies. And of course, I need to thank you, Hamish and Kevin, for giving me the chance to speak here today. It's been great. Thanks. I'm happy to answer any questions now.
Thank you so much, Charles.
Thanks, guys.
Now, if people want to type in a question into the Q&A box, we can go through it. You can try like in the chat window too, but better to go in the Q&A. And while we're waiting for people to write their question, I might start, if that's okay.
Go ahead, mate.
So you're talking about how there's this hydrophilic and hydrophobic part. How do fats pass through cell membranes then? Do they get stuck in the middle because they want to be in that hydrophobic part?
Yeah, that's an excellent question. And often they do. So if you consider, there's various hormones like estrogen, for instance, which is, it's a sterile hormone. And it's got to get from the blood into the cell nucleus. Now, the good thing about it is that it can cross into the cell membrane because it's a sterile, a bit like cholesterol is, and it can get in there. But then it needs to also get out, go through the cell, and get into the nuclear, through the nuclear membrane as well, in order to get to its receptors to do its actions. And these things, that being amphiphilic, they have a certain on rate and a certain off rate. So yes, they can get into membranes, but they can also get out again, because they've got two portions, right? They're hydrophilic and they're hydrophobic. And if they bunch together and they form in a certain way, then maybe they're happier in water. But if they split up, maybe they're happier in the membranes. But it's not an either or. It's I can go in and I can come out again as well. So certain molecules have the ability to be like anesthetics, happy in both. Yep. More potent anesthetics like to be in membranes. That's just a correlation that those two scientists found. But they can happy to be in both. Excellent question.
Okay, we've got a comment rather than a question, which I'll read if the attendee doesn't mind. Watching Acosta live stream made me love gardeners. Watching this makes me love scientists. Hoping to study biochemistry and botany or immunology next year, and psychology as well. So much still to uncover with our understanding of how we work. Thank you for the talk.
Thank you, Dan.
I'm just going to keep asking lots of questions. Kevin, do you have any questions? I'm going to be one of those annoying chairs that ask questions. Can you comment at all on the difference between the regular cell membrane and the nuclear cell membrane, or whatever the nuclear membrane is?
No. Sorry, I know it's made of lipids, and it's probably, there could be certain lipids there might be more of in the nuclear cell membrane, but I actually haven't looked into that.
Yep. That's
Sorry.
okay.
Sorry. I know cells, when they undergo apoptosis, what happens there is that on the outer...
That's cell death, right?
Programmed cell death, yeah.
That term, yeah.
Apoptosis, yep, programmed cell death. So I know in those cells, you get all of a sudden you see a lot more of one particular type of lipid, a phosphatidylserine lipid, go onto the outer leaflet of the membrane. So whereas it might have been on the inner leaflet, all of a sudden it's being transported to the outer leaflet of the membrane. No one knows how it gets transported. There are things called, there are enzymes called Phospholipases, which like to flip lipids from one side to the other. Some people think they might go through pores that might get created in the membrane. But when their cells undergo apoptosis, you get a lot more of this particular one lipid called phosphatidylserine. And that's how we can tell if a cell is apoptotic. We've got antibodies to this particular lipid, and we can then label those cells and say, you're a cell undergoing apoptosis because you've got a lot of phosphatidylserine in your outer membrane. Cancer cells also have a lot of phosphatidylserine in their outer membrane as well. Yeah.
Okay, thank you. We've got a question from Raymond. If it was 1972 when that cell wall came out in Science, how long until it appeared in high school textbooks?
Okay, good. Well, as Hamish pointed out to me today, we've got to be careful about cell wall and cell membrane. Cell wall is what plants have, and it's made of cellulose. And underneath that cell wall in plants is the cell membrane. And so I shouldn't have labelled my talk the way I did, calling it cell wall.
Our attendees can handle it. All good.
Oh, good. So that particular model of a cell membrane, the membrane mosaic model, did come out in 1972 by those two scientists who I mentioned. And how long until it appeared in high school textbooks? I don't know, but it's in almost every high school textbook now.
It was in there when I was in high school, which was in the early 90s.
Yeah, well, I was only born in 1971, and it was in the high school textbooks there. I think leading up to it, there might have been a lot of discussion about that anyway. I think what happened before that was there was also a model where they had a cell bilayer plus a protein layer. So it was like a 3-layer model or something. Or there might have been a lipid layer with a protein layer and another lipid layer. I can't remember now. Because they knew that there were proteins around about the place at the same time. Electron microscopy could say, oh, there's proteins there as well as lipids, but their resolution wasn't good enough to decipher what was what. Sorry, there's a phone going on in the background in my house. I apologize for that.
That's okay.
So there wasn't conclusive. But then these guys came up with the lipid mosaic model, and all the data seems to fit that model so far. And electron microscopy is getting a lot better as well. The resolution in electron microscopy is getting a lot better. So we can... Yeah,
I encourage our audience to Google some electron micrographs or electron microscopy images. They're often quite amazing, spectacular. We've got another question from Mafalda, who says, you just said cancer cells have lots of phosphatidylserine. What other molecules are common on the outside of cancer cells?
An excellent question. And it's not all cancer cells that will express phosphatidylserine. There'll be some cancers, not others. Cancer is a description of so many different pathologies when it comes to cells and different types of cells that you can't really classify all cancers as one. And there are some proteins and some enzymes as well that are specific to many cancer cells as well. So they can also be targets as well. And that's what a lot of drug companies are looking at. They do huge screens now where they get cancer cells and they try a whole suite of molecules, and they've got robots doing it. These ones are dying, these ones aren't. Okay. And what they also need to do is test them against healthy cells as well to make sure that they're targeting cancer cells better rather than other cells.
Thank you. Another question from Raymond, who's asking, is it possible to wrap other things like an active constituent inside a membrane for delivery to a site?
You know the answer to this already, Raymond. Yes, of course you can. And those, they're called liposome nanoparticles. And there's a lot of research right now into liposome particles, where they load these particles up with a particular drug. And in the membranes of those liposomes, they can maybe put a protein or an antibody that's going to target somewhere. And when it gets to its site of action, then something else happens and it breaks open the liposome and the toxins come out and kill all the cancer cells. And so there's lots and lots of, if you go through the literature, there's lots of these nanoparticle research papers now where they're trying to target certain sites using these little carrier packages. Yeah, good question.
Okay, we're getting lots of questions now, but so the next one is from Christopher. If you put the right materials together, can you get cell membranes or similar materials to form spontaneously, suggesting how cells were first formed? Do they do it more easily in extreme environments, a la extremophiles?
That's an excellent question. I really like this one. So when I make those tethered bilayer lipid membranes, I'm actually relying on self-assembly. So we get our lipids, and we've got these tethering molecules on our gold electrodes, and we get lipids, and we got them dissolved in ethanol, and they're just floating around in the ethanol around our tether molecules with their hydrophobic tops. They're just floating around, and then we wash through some water solution, an aqueous buffer, a water solution, and the lipids just want to go run and hide, and so they self-assemble around our tethering molecules and form a bilayer for us. This happens almost instantly. It takes us less than 15 minutes to put one of these TBLM slides together. So we absolutely rely on self-assembly, and lipids do self-assemble. It's an excellent question. Now, if we go back way back in time to evolution, and I've only done a small bit of reading about this, there were hydrocarbons there, which could have been lipid-like, and we know this from the Murchison meteorite that hit Earth back in the 1970s, Murchison in Victoria, where I'm from. Hello to all my family in Victoria. I hope you're coming out of lockdown soon. But the Murchison meteorite, they looked at it and said, oh, look, there's amino acids in here. Oh, look, there's long chain carbon molecules. Oh, these could be the early building blocks for membranes as well. And so back in that primordial soup, there were mostly single-chained amphiphiles that could create a kind of a membrane for the evolution of life. Now, what was the rest of the question? I've forgotten it already.
If you put the right materials together, can you get one forming spontaneously, suggesting how cells are first formed? Do they do it more easily in extreme environments?
Do they do it more recently in extreme environments? That's an excellent question as well. Yes and no. If you remember that shape I was talking about, like some lipids have certain shapes. Well, when you put them in extreme environments, like really low pH, for instance, that also affects their packing. And this is something that came out of our lab, actually, so we're very excited about this, because the pH is a hydrogen proton that that makes things acidic. But when it mixes with water, it becomes something called a hydronium ion, which is actually called H3O+. But when it gets to two lipid head groups, and you put the H3O+, the hydronium ion there that lowers the pH, it actually brings these head groups together and it packs them more tightly. And so it could be that under extreme environments, you might be more likely to create those membranes. And also, we've noticed that effect, when you pack the head groups together, the tails have to get longer in order for it to maintain its conical shape, to maintain its membrane shape. And so in extreme environments, in extreme pH environments, you often get lipids with shorter tails. Whereas in high pH environments, you get lipids with longer tails. And that could be caused just because of the packing in order to maintain that membrane shape. That's our model. We're sticking by it. There's a great paper in the literature about it.
There's another question about shapes, but I'll get to that in a moment because there's a few other ones. Tibor asks, cells die and are replaced all the time. Are the lipid molecules recycled or reused, or are they disposed of and new ones extracted from food, etc.?
Oh, it's highly dynamic. So yes, they're reused. Yes, they're used for food. So one of the common, you can store only a small amount of glucose in your body, and you can store glucose, which is a sugar, as a polymer in your body in something called glycogen, but you can only store about a tiny bits in your liver and in your muscle cells, if you like. And it's only, I can't remember now, 0.2 grams or something. It's a tiny amount that you're able to store. However, you have an unlimited ability to store fats. You can just keep on getting bigger and bigger and bigger, and I'm working on it. I'm working on it big time. And that's good. That's important because you need to be able to store fat in order to burn it and to eat it. And you break down those fats and it goes into the citric acid cycle or whatever, it becomes as acetyl-CoA. And you burn those fats and you need it for energy. And it's a great source of energy because it's got so much energy in there. And so your lipid membranes can then be, and there's enzymes there to change those phospholipids into triglycerides and other forms of fatty acids that you can put into storage once they're used or they're not needed anymore. So yes, it's backwards and forwards. We're breaking them down, we're converting them, we're reforming them. It's all happening all the time. Yep.
Thank you. Okay, another question from Mafalda who asks, can you comment on immunological medications? A huge amount of medications coming out.
I can give enough. I guess you're talking about cancer here, because there's specific monoclonal antibody therapies for cancer cells and things. And they're very, very good, but your body does create an immunity to them eventually. So they've only got a short lifespan. And so when it comes to cell membranes, though, I can't really comment too much about them.
That's okay. So back to the question about shapes. Raymond was wondering, out of the cone and cylinder structures, whether one is more common than another.
And certainly in cell membranes, they're conical. Otherwise, you don't have a cell membrane. Your cell membrane starts to fall apart. You can that's why detergents work to break open cell membranes, because all the hydrophobic parts from the detergent, those single conical ones, they all want to go into the bilayer, and they just change the shape of the bilayer such that the bilayer will start forming micelles. It depends on the concentrations of each. So in your body, when it comes to cell membranes, you certainly want more cylinders than you do cones. There might be cones in there, and they can change the structure of the cell membrane, make it more or less fluid, but overall you want more cones. In nature, though, I'm sure there's detergents in nature and things as well. I can't think off the top of my head what the proportions there are out there overall.
Thank you. No worries. Any other questions people want to type into the Q&A box?
We've got an extra one there.
I think we answered that one already.
Okay, yep.
Yeah. Thanks, Kevin. Okay, well, look, we might wrap it up there then. Thank you so much, Charles. Thanks to everyone who dialed in. Really appreciate you taking the time on this fascinating topic. There's clearly a huge amount of research happening in it, so all the best with it, Charles.
Oh, thank you. Thank you, Kevin. Thank you, Hamish. And thank you, everyone, for attending. If anyone's got any more questions, they're welcome to send me an email.
Yeah, get in touch with Charles directly or get in touch with us and we can pass the question on. And as Charles mentioned, thank you. We're recording this, so you'll be able to access it afterwards. I'll just wrap up by thanking our sponsors, Inspiring Australia and the New South Wales Government, and looking forward to seeing you at our next event, which is, remind me, Kevin?
15th of November.
15th of November, yes, with Stephen Camper on pain. So that should be a really interesting one too. Thanks, everyone, and we'll see you all soon.
All right. Bye.